We are going to talk about the DNA replication. Our team consists on the participation of Moreno Lares Jesús Emiliano, Toral Rodriguez Sharon Belinda, and Zaldívar Villagomez Daira Elizabeth. The ability of a cell to survive and proliferate in a chaotic environment depends on the accurate duplication of the vast quantity of genetic information carried in its DNA. This duplication process, called DNA replication, must occur before a cell can divide to produce two generically identical daughter cells. Maintaining order in a cell also requires the continual surveillance and repair of its genetic information, as DNA is subject to damage by chemicals and radiation in the environment. Each strand of DNA double helix contains a sequence of nucleotides that is exactly complementary to the nucleotide sequence of its partner strand. Each strand can therefore serve as a template or mold for a new synthesis of a new complementary strand. DNA strands S and S' can serve as a template of making. Strand S can serve as a template for a new strand S', while strand S' can serve as a template for a S. In about 8 hours of dividing animal cells, we'll copy the equivalent of 1,000 books.
Adenine and guanine are purines. Purines are the larger of the two types of bases found in DNA structures are shown. The 9 atoms that make up the fused rings, 5 carbon, 4 nitrogen, are numbered that 1 to 9. All the ring atoms lie in the same plane. Cytosine and thymine are pyramids. The 6 atoms, 4 carbon, 2 nitrogen, are numbered 1 to 6. Like purines, all pyrimidine ring atoms lie in the same plane. Cytosine forms 3 hydrogen bonds with 1 ion. Thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5', 3' direction relative to the hydrolyx group of the deoxyribose sugar and the other strand oriented. In a 3' to 5' direction, this shows the antiparallel. Nature of the DNA strengthens the complementary base pairing. In the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template of the creation of a new double strand. Replication begins with a double-stranded DNA being separated on each original strand. Called a parent strand is used as a template for a complementary base pairing of nucleotides to make. 2 new molecules DNA replication occurs in the 5' to 3' replication. So new nucleotides are adding to the 3' end to the newly forming strand. DNA replication will begin at a specific area of the molecule. called the origin of replication. The origin of replication denotes the area of the active replication called the replication fork. In the order to understand how complex.
Eukaryotic organisms replicate DNA. Scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed. Once the replication fork is established, helicase separates a strand of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-strand areas outside the replication fork do not supercoil. Once the replication fork is stable, DNA's polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins such as beta-clamps and the clamp loader help to hold the DNA polymerase in place on the DNA. Short sequences of RNA called primers have to be paired on the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as a leading strand. New nucleotides are added to the pre-prime end, moving continuously towards the expanding replication fork. Discontinuous synthesis occurs on the parent strand oriented to 5' to 3' called the lagging strand, and is complemented in segments called Okazaki fragments. Replication on its strands uses primers to add primers ahead to the 5' end on the lagging strand. DNA polymerase number 3 adds short sequence of nucleotides, the Okazaki fragments to the primer filling in the gap. As the helix is opened further, this process repeats until the starting strand is replicated. DNA polymerase number 3 replaces the RNA primers with DNA nucleotide, and DNA ligase is used to ensure bonding between the fragments and replace the nucleotides. Once the leading and lagging strand have completed their replication, two identical copies of the DNA result. The process of DNA replication allows actively dividing bacterial cells to make sure all cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. This bacterial population can grow. Next, we're going to put a video of an animation about the DNA replication. I hope you enjoy.